behind it, that traveled over 100,000 miles and visited more than 4.5 million flowers. A very good afternoon, one and all. Myself, Shashank from 2nd BZC. On behalf of the Nature Club, I hereby welcome you all to the program Know Your Bees Better, conducted by Nature Club in association with Department of Zoology, Department of Botany and Internal Quality Assurance Cell. It is no wonder that many great men and women who have been inspired to study these beautiful creatures and write amazing phrases, poems and songs on them. It is not just about their beauty and inspiring hard work, but they are also the essential part of the existence of many other life forms. A small cheer and a great welcome can make a merry feast. I would like to call upon Miss Suchitra from 2nd BZC to formally welcome the gathering.
A very good afternoon to everyone. Today we all have gathered to enlighten ourselves by listening to the guest lecture on Know Your Base Better. I'm Suchitra from 2nd BZC for privilege to welcome you all to this event organized by Department of Zoology, Botany in association with Nature Club and Internal Quality Assurance Cell. It's my honor to extend a hearty welcome to the resource person of today's event, Sri Suhas Krishna AG, Assistant Professor, Department of Zoology. Welcome you, sir.
Thank you.
I would like welcome you, sir. I would like to welcome IQAC Coordinator, Professor Showprasad sir, to this gracious occasion. Welcome you, sir.
Thank you.
I'm pleased to welcome Professor Sri Krishna Ganraj Bhatt, HOD of Botany Department. I cordially welcome you, sir. I would like to welcome Dr. Smita Preeti, the convener of Nature Club. I heartily welcome you, ma'am.
Thank you, Suchitra.
I extend a warm welcome to all my fellow friends who have joined to us in this platform. Once again, welcome you all. Thank you.
Thank you, Suchitra, for your kind words of welcome. A hearty welcome to you also. We must welcome the future, remembering that soon it will be the past. And we must respect the past, remembering that it was once all that was humanly possible. I would like to request Ms. Sushmita Bharadwaj from 2nd BZC to take this forward with an introduction about the resource person.
The key to pursuing excellence is to embrace an organic, long term learning process and not to live in a shell of static, safe mediocrity. Usually, growth comes at an expense of previous comfort or safety. A very good afternoon, one and all. Myself, Sushmita from 2nd BZC. We are excited to have you all for the talk Know Your Bees Better by the resource person Mr. Suhas Krishna AG. He is from Nekkiladi near Uppinangadi. He completed his post-graduation in applied zoology from Mangalore University. He is a trained beekeeper, received training from SK Beekeepers Cooperative Society, Puttur. Since the past 8 years, he is working as a lecturer in zoology at Vivekananda Degree College, Puttur. Once again, I am delighted to offer you a very cheerful welcome, sir. Thank you.
Thank you, Sushmita, for introducing the guest. The men of experiments are like butterflies. They only collect and use, but bees collects materials from the flowers of gardens and of the fields and convert it into honey by the power of its own. Now I request Suhas Krishna sir to address the gathering. Over to you, sir.
Thank you, Sashank. Good afternoon, everyone. Today I am going to talk about bees, so honey bees. So right away I will start showing you the PPT. Can everyone see the screen?
Yes, sir.
Okay.
Yes, sir.
Okay, then no problem. We'll start it. So know your bees better. It is the topic of today's talk. So first of all, I would like to introduce about honeybees. So honeybees, if you are rearing honeybees or cultivating them, then it is called as apiculture. So apis, it is a Latin word. So the meaning of this apis is bee. So apiculture means you are taking care of honeybees. So why we are taking care of honeybees? Why we are looking after honeybees? So there is a aim behind it. So in order to get several bee products. So honey is one of the bee product, but other than honey also we are getting so many other products like beeswax, pollen, then propolis, and royal jelly, etc. So there are so many bee products which are of importance. So these honeybees, they are very interesting creatures, okay, and they belong to genus Apis. So the name of the genus is Apis, and there are several species of honeybees under genus Apis. So the special quality of these honeybees is that they are storing honey. So no other insect other than honeybees, they are storing honey. Okay, but these honeybees are storing honey, and they are one of the important insects which are useful for human beings. So another interesting characteristic of these honeybees is that they are building combs or hives using wax. So this particular wax is called as beeswax. So these honeybees, they belong to class Insecta and belong to subfamily Apinae. Okay. So how does the bee look? What does a bee look like? So if you want to identify a honeybee, then you have to look for several characteristics. So honeybees, they are oval-shaped creatures and light brown in color. So somewhat light brown and yellowish in color. And you'll find stripes of yellow and black in the abdomen region. So if you find all these colors, and if you find head, thorax, and abdomen region with the wings, so then you can say that it is a bee. So as you can see in the picture, it looks something like that. So why these bright patches are there or white stripes are there? So they are there because to just to give warning to the predators. So if there are any predators, then these predators will come to know that there is a honeybee and it is having the capacity to sting. So something dangerous is always showing bright colors. So that is the thing of nature that you can see. Not only in honeybees, in other animals also you can see these bright colors. So coming to anatomy. So if you are good in identify the honeybees, then you have to go into the body. So if you are entering the body of the honeybee, then you will find some parts, organs inside the body of the honeybee. So the body of the honeybee can be classified into three regions, okay? Head, thorax, and abdomen region. So in the head region, you will find brain, okay? Brain, pharynx, and glossa, important parts. Okay, and in the thorax region, you will find the wing muscles, air sacs, and aorta. Okay, so salivary gland is also coming in the thorax region. So if you come to the abdomen, then you will find honey stomach, then small intestine, rectum, poison sac, and sting. So some of the important parts that you find in the abdomen. So you can see the honeybee, it is going to flower and collecting the nectar. So whenever it is sucking the nectar, this nectar enters the glossa region. So glossa is nothing but the tongue of the honeybee. Okay, another name for the tongue is glossa. So it sucks the nectar from the flower, and that nectar enters the glossa, and then it will go to the esophagus, and then it will go and get collected into the honey stomach. So it does not move further. Okay, it does not move further, and it will be collected in the honey stomach. So there all the nectar will be collected, and then this honeybee, it will start flying, and then it will go towards its nest. So while it is doing that, slow chemical process starts taking place. So in the honey stomach, the nectar will slowly start converting into honey because some chemical reaction will start taking place. So you will find some enzymes like invertase inside the honey stomach. So this enzyme, it will start converting the nectar into honey. So it is a very slow process. So this happens, it starts happening. So later when the honeybee reaches the honeycomb or the hive, what happens? It will go and it will regurgitate, or it will make the nectar to move back into the cells or honeycombs. Okay, so now what happens? The nectar will be converted into honey, not complete honey, but a little bit of honey. And later again, after storing it in the honeycomb and then fanning it and removing the water vapor from it, then only it will be converted into honey. So all these are the important glands and also parts of honeybees. So you can see in the head region, you will find hypopharyngeal gland. Okay, hypopharyngeal gland is present in the head region, and this is the gland that is secreting this royal jelly. Okay, so royal jelly is a special type of jelly that is given to the developing larvae of the queen. So the queen larvae, it will feed on this royal jelly and it will develop into a queen. So if royal jelly is not given, then that larvae will develop into ordinary worker. Okay. So other than that, near the head region, you will also find this labial and post-pharyngeal gland. Okay, so this is important for the secretion of beeswax. So then in abdomen region, you will find nasonaph and tarsal glands. And then in the thorax region also, there is another wax gland that produces beeswax. So then other than that, you will find other parts like bee pollen. So in the hind leg, you can see this pollen load. So the honey bee, it will go to the flower, and while after collecting the nectar, it is also collecting pollen. Okay, so the pollen will be stored in its hind leg, and it will be stored in the form of a load. Okay, so that is called as a pollen load. So why these bees are collecting the pollen? So pollen acts as a protein source for the bees. So that's why they are collecting the pollen. So next we will talk about the caste of the honeybees. So inside a honeybee colony, mainly there are three types of honeybees or three castes. So first one is worker. So in the diagram or in the picture you can see the worker. So worker bee. So in a colony of about well good or nice population colony, which will have about 20,000 to 50,000 workers. Okay. So then queen also, in the picture you can see the queen. So this queen is somewhat different from the worker. So you can see the body length, it is larger. So the queen looks larger than the worker, and also the coloration is a little bit different than that of the worker. So other than worker and queen, you can find another one, that is a drone. So drone is the male honeybee, and queen is the female honeybee. Then you might have one doubt. You might have one doubt. What is this worker? The worker, it is a sterile female. Okay, worker, it is a sterile female, and it is not able to reproduce. Whereas the queen, it is the only one honeybee in the colony which can lay eggs and reproduce. Okay, so drone is the male honeybee. Okay. So here also in this photograph you can see drone, queen and worker. So A is the drone, B is the queen, and C is the worker. You can see the difference. So male is somewhat darker in color. So abdomen is darker and it is a little bit roundish, globular. Whereas in case of queen, the abdomen is not black, it is somewhat orangish, and it is longer. The abdomen is longer. And this worker, it is for medium size, in between a drone and a queen. So here another photograph, it is there, and in this photograph there is a queen. So can anybody say where the queen is? So this is left for you to identify.
The large
Can any
one.
student? Yes, the larger one. What is the position? Is it in the above or is it in the middle?
Below.
Yeah, below, in the nearly the middle position, right? So that is the queen. So you can see it is the queen of Apis cerana. Okay, Apis cerana species. So you can see the thorax region. Thorax region is darker. So if you compare the thorax region of queen and worker, the thorax region is darker. The queen's thorax region, and also the abdomen region of the queen is very long. So the difference is very clear. So if you are able to identify the queen, then it becomes very easy to do beekeeping. So bee stages. So the developmental stages. Okay. So as I told you before, there is a worker, there is a drone, and also there is a queen. Okay. So the queen, the egg of the queen, it will be for 3 days. So after laying, it will stay in egg stage for about 3 days. So later, it will become larva, and it will be there like a larva for 5 days. So after that, it will enter the pupa state, and it will be there for 9 days. So total it takes 17 days or 16 days for the queen to become adult and come out of the pupa. So whereas worker, the egg is 3 days, larva 6 days, pupa 12 days, and total 21 days. And drone, it is 3 days, larva 7 days, pupa 14 days, and total 24 days. So you can see there is a difference in the development, number of days taken for development. So species of honeybees. So you can see this is Apis dorsata, or it is commonly called as rock bee. So usually this species of honeybee, it is found in open areas. Okay, it likes to be outside, and it likes sunlight. So another interesting thing is it builds a single comb. Okay, large single comb it builds, and it prefers to live in high regions like a very giant tree, or in the buildings, or under the bridges and all. So in such places you will find this Apis dorsata. So the thing is, you cannot cultivate this species of honeybee, although it is producing a lot of honey, and you can get up to 30 kilograms from one hive. But the problem is it does not stay in one place. So because of that nature, since it is not staying in one place, it is migrating from one place to another place. You cannot cultivate this honeybee. Okay. So next one is Apis florea, the little bee. So this, in this species, the thing is, it is also building a single comb, but very small. Okay, very small, and the size of this individual bee is also very small. Okay, and the production of honey is also very less. So it may produce like 500 ml in 1 year. So that is the production of this honey by the Apis florea. So Apis cerana. So Apis cerana is the bee, honey bee that we are able to cultivate inside the box. So using the bee box, you can cultivate this Apis cerana. It is also called as Asiatic honeybee, and it is native to our India also. So here, whatever you find in South India, it is nothing but Apis cerana, the cultivatable species. So Apis mellifera. So this is the next one. So this is also very similar to our Apis cerana, but the thing is, it is not of our origin. It is not coming from India. It has its origin from Europe. Okay, so that's why it is called as European honeybee. So this Apis mellifera, it is slightly larger than our Apis cerana, and it also produces a large amount of honey. So the production ability of this Apis mellifera is more than that of our Apis cerana. So it is producing about 30 kilograms or 40 kilograms per year. From one hive, you can able to get that much. But Apis cerana, you can only get about 5 kilogram or 10 kilogram from one hive in 1 year. So this Apis mellifera also builds several combs. Okay, it is not like Apis dorsata or Apis florea. This Apis mellifera also builds multiple combs. Okay, you can see in the diagram in the photograph. So it is building like that, and our Apis serena also is building multiple combs. Okay. So next one is Tetragonella species. So this is also called as stingless bee or dammer bee. So this stingless bee or Tetragonella species, this is called as stingless bee because it does not sting. Okay, does not have the ability to sting. So the sting is absent. Okay, and also the size of this honeybee is very small. Okay, so very small bees, and it is building its colony inside a very dark place. So it prefers to build inside some hollow of a tree or inside cavities or any other small areas where there is no sunlight. So in such areas, these Tetragonella species or stingless bees are building their colonies. So these are also cultivable, just like Apis serena, Apis mellifera. These Tetragonella species or stingless bees are also cultivable. You can cultivate them. So if you have a small box, you can transfer their colony into that box and you can cultivate them. But the thing is, it produces very less amount of honey. So in 1 year, you may get up to 250 250 gram to 1 kilogram. That much, only that much you can get. But the thing is, the cost of this honey, since it is very less, the cost of honey is very high. So for 1 kilogram of this stingless bee honey, it is about 2,000 to 3,000 rupees. Whereas our regular honey, local beekeepers, they are selling it for rupees 500 per kilo. And if it is, if you are going for this company honey, it is like 250 rupees or 300 rupees. Because it is not a pure honey. They are mixing all these preservatives, other things and all, and it is not a pure honey. Whereas if you want a pure honey, you can ask for any beekeeper and you can get it. But the cost will be 500 rupees. So next, if you want to cultivate honey, then you have to know how to take care of honeybees. Okay. So that for that you have to do apiculture. So how to start apiculture? So for the beginners, you have to get one colony of honeybee. Okay. So the colonies are available in near these beekeepers. Okay, you can contact any beekeeper and you can get the colonies. Other than that, you can also collect honeybees and from the wild. Okay. So sometimes what happens, all these honeybees, they are migrating. So one colony will be there, and from that 50% of the honeybees, they will come out and then they will start finding another place. So this event is called as swarming. They will fly and they will keep searching for new place. So such a swarm, if you are getting, then it is easy for you to catch them and put them into your bee box. Okay. So just like in the photograph, this is how the swarm of honeybees will look like. So while they are flying, you can see this the left side photograph. It appears like this. A swarm of honeybees are flying, and in the right side photograph, you can see they have settled down. So they will come and sit on the, or hang on the branch of a tree like this. So this is another photograph. So here in these photographs, you can see all the honeybees they have gathered. A swarm, this is called as a swarm of honeybees. So you can catch all these bees slowly, and then just using your hand, you can catch them. Or if you are not able to do that, you can use gloves, and slowly you can grab these honeybees gently and then put them into the box. Okay. Once the queen bee, if you get the queen bee, then what will happen? And you will get the control. So slowly, if you get the queen bee and put the queen into the bee box, what will happen? All the other honeybees, they will start entering the box. So this is another method to capture the honeybees. So natural hives, if you find any natural hives of honeybees, it is possible to catch them. So this is one colony of honeybees inside a termite mound. Okay. So this is how it will be appearing, and you can slowly cut the combs one by one, and then fix the combs into the frames of bee box. So after fixing all these honey combs into the bee box, then you can do the same procedure that you done for the catching of the swarm. Okay. So you have to catch the honeybee, and you should get the queen, and then all the honeybees will come into the box. So these are the traditional hives. Okay, so they are using pots and bamboo in order to do apiculture. So this is the modern beekeeping method in which you are using modern bee box or hive. So all these are the parts of bee box. So the topmost thing that you can see, it is called as outer cover. Okay. So below this outer cover there will be inner cover, and below this inner cover you can see honey super. Okay. So honey super is the chamber inside which you can find super frames. So inside these super frames only honey will be stored by the honeybees. Okay. So below super, honey super, you can see queen excluder, and below queen excluder there is a brood chamber. So brood chamber consists of frames which will have combs filled with larvae and eggs. Okay. So the queen will be present in this brood chamber, and it will keep on giving or laying eggs. Okay, and this queen excluder, it separates the queen from entering the super. So that's why the queen cannot enter the super. Okay, so because of that, what happens? All the honeybees, they will store the honey in the honey super, and they will not store much honey in the brood chamber. So majority of brood chamber will be filled with eggs and larvae. So below this brood chamber, you will find a bottom board. So bottom board is present, and below that bottom board you can place a stand. Okay. So all these are the parts of a bee box or hive. So this is how a place with the bee box looks like. So it is called as apiary. So if you place several honeybee boxes or hives, then that place is called as apiary. So during rainy season, you can protect the box with the help of a plastic sheet like this. So you can see below the box, you can see that stand. So this is a typical stand that is used in our place. So in Dakshin Kannada and all, you can see such kind of stands, which is made up of iron. So this iron stand, it protects the bee box from ants. Okay. So some people, they are applying grease to the middle of this stand. Okay. Some people are also applying mud oil to the cloth, and then tying the cloth to this stand, so that no ants will come up and enter the hive. So in the rainy season, if you protect this hive using this kind of plastic sheet, then water won't be going into the box. So inside the brood chamber, there is a brood frame. Okay, so this brood frame somewhat looks like this. So at the base of this brood frame, or at the top of the brood frame, you can see that foundation sheet. Okay, so foundation sheet is present, and just below that foundation sheet, you can see the comb that is built by the honeybees. Okay, so if you look at the bottom, okay, if you look at the bottom of this comb, you can see some cells, okay, round peanut-like cells. So those cells are called as queen cells. So when the colony becomes larger and larger, the colony decides to make one more queen or new queen. Okay, and what happens? The honeybees or the workers, they will start building this queen cells. So inside the queen cell, the old queen, it will come and lay the egg of the new queen. Okay, and that egg will start developing into larva, and that larva will be fed by royal jelly. Okay, so that larva will feed on royal jelly continuously for about 16 days, and then it will emerge and become a new queen. So this is how it looks like, the queen cell. So before the initial stage of queen cell is called as queen cup. Okay, so inside the cup, in the right side photograph, you can see the developing larva. So inside the queen cup, you can see the developing larva of the queen, and also royal jelly is present around that larva. So here in the upper photograph, you can see there is a brood chamber frame. So brood chamber frame looks like this, and below you can see this photograph, which is a honey chamber frame, okay, or super chamber frame. So in the super chamber frame, you can see it is filled with the honey, okay, completely filled with the honey. And in the brood chamber frame, you can see that comb is filled with the larva and eggs. So this is me holding this brood chamber frame. So it looks like this. So you can see the left photograph. I am holding this brood chamber frame, and it is filled with the larva and honey, and also honeybees are there. They are taking care of these larvas and eggs. So the right side photograph, so it is nothing but a super frame. So super frame is filled with honey only, nothing else. So all these are the super frames. I have kept it back to back, and it is filled with honey. Okay. So this is how a typical bee box looks like inside. Okay. So you can see all these are the super frames. So totally there are 8 such super frames. Okay. So I have removed few of them. So if you count, there are 1, 2, 3, 4, 5, and 6. 6 frames are there. 6 super frames are there. I have removed 2. And below these super frames, you can see that brood frames. So one brood frame is visible. So this is how it looks like. If you open the bee box or hive, you can look, and if you see, you can see like this. So this is how the eggs appear. So I told you, in the brood frame comb, you will find only eggs and larva. Okay. So if you zoom into that, inside these cells, each cell is hexagonal. So hexagonal cells. So inside the hexagonal cells, you can see small rice-like structures. So rice grains-like structures. These are nothing but eggs of honeybees. Okay. So all these eggs are laid by the queen. Okay. The queen will slowly come and start laying. So it will lay the eggs exactly in the center of the hexagonal cells. Okay. So slowly the eggs will start developing into larva. So in the upper side of this left side photograph, upper side you can see the eggs have developed into larvae. So you can see the larvae also. So in the right side photograph, you can see the honey that is filled inside the super frame. So inside the super frame, it is filled with honey, nothing but only honey. So next I will show you one video. Okay, the video is not playing, I think. It's okay. So this is how the honey looks like. So after extracting the honey, after filling the honey into bottles, this is how the honey looks like. Okay. So can anybody say the difference in these bottles? What difference do you find?
So color is varied, sir.
Yeah, there is difference in color, right? All
Yes, sir.
these honey, they are of different colors. So the thing is, each of the bottle was like honey was extracted during different months of the year. Okay? So February, the leftmost bottle, okay, that bottle consists of honey that is extracted during February month. So the color appears like that. So if you take extract honey during March, then the second bottle, that is how the honey color will be. The third bottle is April. So April month's honey. And then last one is May month's honey. So you can see the difference in the color. So can anybody say why there is a difference in color of honey? Can anybody guess why the color is different? Why the honey color is not same?
Because there is time gap, sir.
Okay, time gap. So what happens if there is time gap? So during the different months of the year, different flowers will be blooming, right? So when there are different flowers, the nectar that is coming from these different flowers will have different colors. Okay, that's why you will find the color of the honey is different. Okay, so during February and March, you will find most of the rubber plants, they are yielding the nectar. Okay, rubber plants. So the color of the honey will be like this. Okay, because the nectar is coming from the rubber plants. So after that, you will find some of the flowers coming from this kuntala. I think you know what is kuntala. So from that plant, most of the nectar is coming. So especially in this Dakshina Kannada region. So that's why you will find different types of colors and different nectar coming from different months. So next we'll discuss about parasites and disease. Okay. So in the left upper photograph, you can see the comb is infected by this wax moth larva. Okay, so during rainy season especially, what happens if you are not cleaning the box, if you are not feeding the honeybees with the sugar solution, then this colony will become weak. Okay, and when the colony becomes weak, the parasites or the enemies of the honeybee, they will start attacking the honeybees. So this wax moth, it is one of the enemy of honeybee. It will enter the hive during rainy season and it will lay its egg in the bottom board. Okay, and this egg will develop into larva, and this larva will start crawling towards the comb. Okay, and once it reaches the comb, that larva will start eating the wax of the comb. So that's why the name wax moth, because it feeds on the wax of the comb. So after feeding on the wax, it will start defecating and building some like web-like structures, and it will start destroying the comb. Okay. So once this infection becomes more and more, the honeybees won't be able to function properly in that colony, and they will run away from that place. So it is a loss for a beekeeper if this happens. So that's why it is better to clean the box for every 1 week or for every 15 days. And also you should keep feeding the honeybees during the rainy season, because during rainy season these honeybees are not getting enough nectar. That's why you have to feed them with artificial sugar solution. So other than that, other than wax moth, you'll also find these mites. So mites are also found and on the bodies of honeybees, and they'll start sucking the body fluid of honeybee, and they'll make the bees very weak. So this is another important enemy of honeybee. So this is called as greater banded hornet. Okay. So this greater banded hornet it targets a weak colony of honeybees, and it will go and catch the workers. And it has the ability to kill so many workers in one attempt. So this is also one of the deadliest enemies of honeybees. So one disease. So we can keep talking about diseases of honeybees, but one important disease that is seen in our region, that is called as thigh sac brood disease. Okay. So this, as the name suggests, it is a brood disease. Okay. It is attacking the brood. Brood means the developing larva and egg. Okay. So this disease attacks the brood, and the brood which is infected by this disease, it appears like a sac. Okay. So when you are pulling the larva outside, and if you are holding it upside down, it looks like the sac, just like in the photograph. So that's why it is called as sac brood disease. Okay. And this disease originated from Thailand. Okay. And it originated in Thailand, and it has spread into India also, and in our region also it is seen. So that's why the name thigh sac brood disease. So what are the symptoms? So already told you, it looks like a sac. The brood appears like a sac, and if you see the comb, okay, if you see the comb, you can see punctures on the ceiling of the pupa. So pupa will have ceilings, okay, and you'll see punctures on the ceilings of these pupa, and in some of the cells, the ceiling will be gone, and you can see a pointed structure of the brood. So brood will appear like a pointed structure. So this is one typical symptom of thigh sac brood disease. So this is how you can identify the disease. So if you identify this disease, then the problem is there is no cure or medicine for this disease. So the sad thing is nobody has developed any medicine for this thigh sac brood disease, and the colony, if it has this disease, then that colony will degrade. Okay. So the problem, another danger is the disease will spread to another colony. So if you allow like that only, if you don't do anything, then that disease will spread to another colony. So the worker bees, they will transmit the disease to the next colony. Okay. So that is the problem. So that's why you cannot leave the colony which has this disease. You have to destroy that colony. So usually the brood is burnt. So the brood of the colony which has this disease is burnt. So when you are doing beekeeping, you need some of the equipments and also protection kits. So bee wheel. So bee wheel in the topmost left side photograph, you can see bee wheel. So you can wear this so that the bees are not able to sting you on the face. Okay. So then you can also use gloves. So if you are wearing gloves, they cannot sting you on the hand. Okay. And if you are very scared, you can wear bodysuit. Nobody is wearing bodysuit in India. In Western countries, they are wearing. Okay. So full body bee suit you can use. So there are so many tools used by beekeepers. Okay. So queen excluder is one of those. So queen excluder, it is also called as queen gate. So it does not allow the queen to come outside the bee box. Okay. Then extractor, honey extractor. So this is the honey extractor. So already I showed you about that super frame, right? So after removing the seal of the super frame is placed into this honey extractor. Okay. And slowly you can start rotating the frames inside the honey extractor. All the honey will come out, okay, because of centrifugal force. Okay, and it will flow down through the tap into your container. So this is how you are extracting honey from the super combs. So then smoker. So some of the beekeepers are using smoker in order to make the bees calm. So sometimes the bees get agitated. When a beekeeper opens the bee box, what will happen? All the honeybees, some of them, they get agitated and they will start stinging. So in order to prevent that, you can use smoker. Okay, so smoke has the ability to calm down the honeybees. That is the function of smoke. So all these are the finished products. Okay, all these are the finished products. So honey, then beeswax, then royal jelly. So royal jelly appears somewhat like this. Okay, so inside the queen cell, the larva will be there, and around the larva, queen larva, the royal jelly will be present. So by feeding on the royal jelly, the queen larva will develop into an adult queen. Okay. So this royal jelly has a special ability. So that's why humans are behind it. Okay. So some of them have some misconception that if you are regularly consuming royal jelly, then you will also become like a queen. That is a misconception. Although it might be good for health, but you won't become something extraordinary. Okay. So that's why this royal jelly has demand in market. So beekeepers are removing the royal jelly also, and they are selling the royal jelly in terms of grams. So thank you. So that was about the apiculture or honeybees. So if you have any doubts or questions, you can ask.
Thank you, sir, for your enlightening speech. If anyone has questions, I am sure Suhas Krishna sir will be pleased to answer them.
Okay, one question is there. Why honeybee dies after losing their sting? Okay, this is a good question. So these honeybees, okay, the workers especially, not the drones, the workers, they have the sting in their abdominal tip. Okay, so these workers, they are able to defend their colony whenever a predator attacks. Okay, so if a predator is a human being, so just imagine a human being is opening the or going towards the hive and trying to remove the honey. So at that time, what happens? The colony gets alarmed, and the workers, they will start attacking the enemy. Okay, so some of the honeybees, they will try to sting the predator. Okay, so you can see yourself, your skin. Okay, so it is a very smooth and rubber like, right? So whenever the honeybee comes and stings to your skin, what will happen? The barbs on the honeybee sting, it will enter into your skin, and it will hook into your skin. Okay, and when the honeybee tries to come out or it tries to fly back, what will happen? Because of this hook-like structure, barbs, the sting remains hooked into your skin, and the bee will pull out, and along with the sting, its intestine also comes out. Okay, so because of this process, the honeybee which stings once, it will lose its life and it dies. Okay, so hope you got your answer, Ankita. Okay, so next question is from Preetham. So honey, does it get spoiled even if we preserve it for years? Okay, Preetham, the natural honey. So if you take natural honey, raw honey, and keep it inside a bottle, glass bottle, then it never gets spoiled. Never means never. Like for thousands of years also, it does not get spoiled. If you have kept it in a airtight glass bottle, then nothing will happen to that honey. So this is the specialty of honey. Okay, why it is so? Because inside honey, the water content is very less. Okay, and it is hygroscopic, means it has the ability to absorb water. Okay, So what happens, some bacteria, if it is there on the honey surface, the honey will absorb the moisture out of the bacteria and kill them, okay, because of their hygroscopic property. So that's because of that these microorganisms are unable to spoil honey. So best example is the honey sample has been retrieved from these pyramids of Egypt. So inside the pyramids of Egypt, the Egyptians, they had kept a bottle of honey near their mummy pharaoh and like an offering to their king. And even after thousands of years, that honey is intact and it is edible and it has not gone spoiled. Hope that answers your question, Pritam. Okay, Sushmita Bharadwaj has asked one question. What to do if there are more than one queen bee? Should we separate them? So usually in one colony of honeybee, there is only one queen bee. Okay, so two queens will develop only during splitting or swarming. Okay, when the population of this bee colony increases randomly, and when there is no space inside the colony, then what happens? The colony or the bees, they will decide that they have to divide or they have to split. So at that time only they will start producing new queen bee. Okay, so I already shown you how that queen cell appears and how the queen bee is born. So as soon as the new queen is born, what will happen? That new queen, it will sometimes stay in the colony, and the old queen, along with 50% of the population, it will fly away from that old colony. Okay? So sometimes it is old queen leaving the colony, or sometimes it is new queen, it will leave the colony. So that's why you won't find two queens in a colony. Okay, there will always be one queen. Okay, Sinchana has asked one question. If any adulterant is mixed with honey, then how can we find and how can we separate it? Very good question. So usually all of us as consumers have this doubt. So most of the time, so if you go to supermarkets, you'll get honey of different brands. Most of these brands, they are mixing or they are adulterating the honey with sugar solution. Okay. So that is a problem. That's why all this honey will look alike, same looking, similar color. Okay. So and also they can produce lot of honey by mixing adulterant. Okay, that's why they are able to supply lot of honey. But if you want pure honey, you have to go to the beekeeper. Okay, so if you know any beekeeper near your house or somewhere, you should go to that beekeeper and ask for honey. He will or she will give you a pure form of honey. Directly they will extract in front of your eyes from the honeycomb and they will give the pure honey. So now the question is how to identify this adulterated honey from pure honey. That is an important question. So how can we do this? So there are several tests to identify this pure honey with other adulterated honey. So one of the simple tests that you can do in your house is you can take a glass of water, okay, in a transparent glass water you can take, and then you can pour this honey. Okay, what will happen if it is pure? This honey will slowly travel inside the water and then it will settle at the bottom. Okay, and when it is traveling in through the water, it does not mix in the water. If it is pure honey, it will be like that. But if it is impure, if it is adulterated with sugar solution, what will happen? When it is traveling through the water, the sugar, it will, the sugar solution, it will start coming out of this honey. So you can see that it is not pure honey. So another test is you can use your cotton clothes or cotton dress, anything. So and pour honey over that cotton cloth. So now what will happen? If it is pure, the honey does not stick to the cotton cloth. It will flow like pearl, okay, without sticking. But if it is adulterated honey, it will immediately stick. If it has sugar in it, what will happen? The sugar will immediately stick to the cotton cloth and it does not come or it won't run down. So this is another easy test. Another thing that you can do is a taste. Simple. So if you are tasting pure honey, you'll taste it, its sweetness, and that sweetness will disappear within few seconds, like 30 seconds or 20 seconds. The sweetness will disappear, and you'll consume the honey. But when you are consuming adulterated honey, what will happen? You'll get the sweetness, artificial sweetness of sugar. Okay, and that sweetness will remain in your mouth for about 1 minute. Okay, so at the side of your tongue, at the side of your mouth and all, that sweetness will remain. It is just like tasting sugar candy. Okay, so you can make out the difference. So all these things are, all these tests can be done in order to identify pure honey from artificial one. Hope that answers your question. Okay, next one is Shrisha. How does a swarm of bees choose their queen bee? No, like, see, when the honeybee colony divides, what will happen? A new queen will be already produced. Okay, only after the new queen is born, what happens? That colony decides to split from the mother colony. So in all the swarm of the honeybees, you will find a queen. Okay, so there is no choosing of queen. Already that queen will be there inside the swarm. Okay, so before splitting, before dividing itself, a new queen will be born, and that along with that new queen bee, the 50% of the colony will swarm out of the mother colony. Okay, I think that answers your question, Shrisha. So any more doubts? Okay, Varsha Kumari. Does honey produced by all types of bees get crystallized? Yes, like if you store honey, okay, inside a bottle and keep it for like months or 1 year, 2 years, slowly what happens, the fructose content in that, fructose is a sugar, right? So the fructose content in it will start settling down, or it will start coming to the surface sometimes, and it will start crystallization. Okay, so you will find crystals of sugar. It is not adulterated sugar. Some people have this misconception that only adulterated honey has this crystals. Not like that. Even in pure raw honey, yeah, the thing is raw honey has these crystals. Okay, the processed honey does not have these crystals. So some people, they are processing the honey, they are heating the honey, and also filtering the honey. So if you are doing the heating and the filtering, then there may not be any crystals. Okay, but raw honey, if you keep raw honey directly, if you remove the honey from the bee box and keep it inside a bottle without doing any process, then what will happen? Slowly, as the time goes on, there will be crystallization. Hope that answers your question. Okay, any more doubts or questions?
Any more questions? Otherwise, we go ahead. Thank you, sir, for your enlightening speech. Now,
Welcome.
I would like to ask my fellow friends and express their opinion on the events we have conducted in this semester through Google Meet.
Hello, sir. It's a very wonderful, informative webinar. ಆಕ್ಚುಲಿ ನಾವು ಹನಿಬಿಯನ್ನ ನೋಡ್ತೇವೆ ನಮ್ಮ ಸರೌಂಡಿಂಗ್ ಅಲ್ಲಿ. ಸೊ ನಾವು ಇಷ್ಟು ಇನ್ಫಾರ್ಮೇಶನ್ ಅದ್ರ ಬಗ್ಗೆ ಕಲೆಕ್ಟ್ ಮಾಡಿರ್ಲಿಲ್ಲ. ಈ ಸರ್ತಿ ನಮ್ಗೆ ಪಾಠದಲ್ಲೂ ಕೂಡ ಇದೆ. ಸೊ ಇನ್ನೊಮ್ಮೆ ನಮಗೆ ರಿವಿಷನ್ ಆದಾಗ ಆಯ್ತು, ಅದ್ರ ಬಗ್ಗೆ ಹೆಚ್ಚು ಇನ್ಫಾರ್ಮೇಶನ್ ಕಲೆಕ್ಟ್ ಮಾಡಿದಾಗ ಆಯ್ತು ಇವತ್ತು. ಹಾಗೆ ಇದಕ್ಕೂ ಮುಂಚೆ ಬೇರೆ ವೆಬಿನಾರ್ಸ್ ನಮ್ಮ ಕಾಲೇಜಿಂದ ಕಂಡಕ್ಟ್ ಮಾಡಿದ್ರು. ಮಸ್ಕಿಟೋ ಬಗ್ಗೆ ಆಗಿರ್ಬೋದು, ಅರ್ಥವಾರ್ಮ್ ಬಗ್ಗೆ ಆಗಿರ್ಬೋದು. ಹಾಗೆ ಗಾರ್ಡನಿಂಗ್ ಬಗ್ಗೆ. ಸೊ ಎಲ್ಲವೂ ಕೂಡ ನಾವು ಹೆಚ್ಚಾಗಿ ನೇಚರಲ್ ಅಬ್ಸರ್ವ್ ಮಾಡ್ತಾ ಇದ್ವಿ, ಬಟ್ ನಾವು ಅದ್ರ ಬಗ್ಗೆ ಅಷ್ಟು ಇನ್ಫಾರ್ಮೇಶನ್ ನ ಕಲೆಕ್ಟ್ ಮಾಡ್ಲಿಲ್ಲ. ಸೊ ಇವಾಗ ನಮಗೆ ಫ್ರೀ ಆಫ್ ಕಾಸ್ಟ್ ನಮ್ಮ ಮನೇಲೆ ಕೂತ್ಕೊಂಡು ನಮಗೆ ಇದ್ರ ಬಗ್ಗೆ ತಿಳ್ಕೊಳ್ಳಿಕ್ಕೆ ಒಂದು ಅವಕಾಶ ಸಿಕ್ತು. ಸೊ ಎಲ್ಲೋ ದೂರದಲ್ಲ, ನಮ್ಮ ಹತ್ರನೇ ರಿಸೋರ್ಸ್ ಪರ್ಸನ್ಸ್ ಇದ್ರು. ಸೊ ನಮಗೆ ಕಲೀಲಿಕ್ಕೆ ತುಂಬಾ ಅವಕಾಶ ಸಿಕ್ತು. ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಫಾರ್ ದಿಸ್ ಆರ್ಗನೈಸಿಂಗ್ ದಿಸ್ ವಂಡರ್ಫುಲ್ ಸೆಷನ್ಸ್. ಥ್ಯಾಂಕ್ ಯು.
ನೆಕ್ಸ್ಟ್ ಯಾರಾದ್ರೂ ಒಪಿನಿಯನ್ ಹೇಳೋರಿದ್ರೆ ಹೇಳ್ಬೋದು.
ಇಂಟರಪ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೇನೆ. ಫೈನಲ್ ಇಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಪಾರ್ಟಿಸಿಪೇಟ್ ಮಾಡ್ತಾ ಇದ್ದೀರಿ ವೆಬಿನಾರ್ ಅಲ್ಲಿ. ಏನಾದ್ರೂ ಪಾಸಿಟಿವ್ ಪಾಯಿಂಟ್ಸ್ ಅಥವಾ ಇಫ್ ಯು ಫೀಲ್ ದಟ್ ದರ್ ಇಸ್ ಸಮಥಿಂಗ್ ಲ್ಯಾಕಿಂಗ್ ನೀವು ಹೇಳಿದ್ರೆ ನಮ್ಗೆ ಚೆನ್ನಾಗಿರ್ತದೆ.
Namaste. Good afternoon to all. So myself Anushri Bhatt from third year BSc BZC. So actually ನಾವು ಫಸ್ಟ್ ಇಯರ್ ಸೆಕೆಂಡ್ ಇಯರ್ ಅಲ್ಲಿ ಇರುವಾಗ ಕಾಲೇಜಲ್ಲಿ ಅಷ್ಟು ಆಫ್ಲೈನ್ ಕ್ಲಾಸಸ್ ಇದ್ರು ಕೂಡ ಪ್ರೋಗ್ರಾಮ್ಸ್ ಅಷ್ಟು ನೇಚರ್ ಕ್ಲಬ್ ಇದಾಗ್ತಿರ್ಲಿಲ್ಲ. ಸೊ ಮೇ ಬಿ ಆನ್ಲೈನ್ or virtual platform was like a boon for us. So we got plenty of programs because of this online mode. So I would like to thank for all those who have strived hard to conduct the programs. So thank you for everyone.
Thank you, Anushri, for your opinion. Now I request Sri Krishna Ganraj Bhatt sir to share his view.
Hello everyone. I don't think head of the institution, principal sir is present. I think he is not in the meet. Even IKC coordinator is also not there, I think. So my dear colleagues and student friends, let me share my opinion about the series of programs that we have conducted recently. Hope I am audible. Let me clarify it first.
Yes,
Yes, sir.
Because your network is a bit weak, that is why I have purposefully kept my video off. If I keep it on, it may not reach you, I think, because the network is a bit weak today. First of all, I want to tell you that during this locked period or pandemic period, we tried our best to organize a series of programs. As Anushri said, this time we have arranged a series of programs. Many times I told you that it is just like blessings in disguise. Actually, we don't want to sit inside the house for long. We want to wander here and there. We want to go to college. We want a lively life. But still, the situation made us to sit inside the house. But thanks to the technology, it made us to explore new things. We have learned a lot to share the slides, to prepare PPTs and all, share our views and all, and also to organize valuable programs to our students. It reminds me an age-old saying, all work and no play makes Jack a dull boy. All of you heard of it, I think. I strongly agree with it. I believe that a student should study well, but not focus only on it. Rather, he or she should also participate in extracurricular activities during his or her student life. What is the importance of these extracurricular activities then? Many of you know it, and some of your behavior made me to think that many of you not aware of the importance of extracurricular activities. I can see many of them are leaving the meet now. Maybe due to network issues, or may get bored of. I don't know. I don't value such students if they purposefully leave the meet. These extracurricular activities provide a channel for offering students the opportunity to apply academic skills in a real world context. I mean, these activities are considered as part of a well-rounded education. In my opinion, the extracurricular activities allow the students to pursue interests outside of the academic context. Hope you agree with me. Many of you experienced it. This indeed explore you or expose you to a more diverse range of interests. Some of you might have developed interest towards beekeeping now. Some of you might have developed interest towards cultivating plants so as to rear honeybees, or so as to proceed with a new venture, what is known as apiculture. Definitely what you will learn during this student life, what you will learn as an extra during your student life, that is beyond the syllabus or in support of the syllabus, may help you in your future. I've seen many of them based their future on the hobbies or what they have, what the interest they have developed during their student life. Many of my ex-students excelled well in photography. Many of my senior students excel well based on their hobbies and habits, rather than continuing their future with botany, zoology, or chemistry. They excelled in social work or beekeeping or photography or gardening, etc., etc. Your academics may not help you always. Many times what will help you in future is your interest, your hobbies. To support this, we have conducted, or rather I could say, we tried our level best to organize series of programs. Many of you enjoyed it. Many of you attended it without missing even one. Many of you failed to attend one or two because of technical issues or due to genuine reasons. And so many of others missed it purposefully, thinking that the sir may have some benefits. Mita ma'am may have some benefits. Why should I join the meet? Why should I waste my currency in attending such unwanted programs like that? We offered you opportunities to show your talents, maybe offering vote of thanks, maybe offering or extending welcome notes, or maybe thanking God Almighty or in performing or arranging the program, setting the or arranging the invitations, digital works, etc., etc., or maybe master of ceremony, etc., etc. If you conduct such programs, then only you will have such opportunities to show your talents. Some of you may not know what talents you have. If we give an opportunity, if you agree to, or if you accept to do that, then only you will be able to search or explore your talents. Other day, one student expressed, soon after the program, that student expressed her views that it is because of the opportunity given to her, she was able to what explore what she knows or how to manage the event and all. It is an opportunity or it is a platform for you to improve further in such activities. You can know where you are weak. You can know where you are strong, where you have to improve. Everything you can learn by involving such activities. There is a saying, tell me, I may forget. Show me, I can remember. Involve me, I can understand. I think it is true for many of you students. If we just show or if we just conduct or teach you or conduct a program, you can just witness it. If you are provided with some examples, some visuals and all, it will help you more in understanding the aspects. Instead, if you are made to involve, such as if an opportunity is given for you to present, or if a task is given to you, you will work more towards it. Am I right? While doing so, you will understand it better. So, my concern is, by organizing such programs, many of you would learn more about the aspects. You will develop good ideas so that you can come out with flying colors. I extend my thanks to many of the students who worked hard in organizing these programs. I don't want to mention the names. All of you know who are all on the front line in organizing the programs. It's not enough if we staff members show interest. What is more important is the positive response from you students. If you show such interest, your active involvement, then only we could organize many more such programs. Or if you are also about 60 or 70 students attended today's program, for a few seconds, I could I have noticed about 80 plus students. Within few seconds, many of them left. I think for attendance purpose, some of them attended, no problem. If you are also lethargic, like other students, we may have to stop these programs in future. One thing I can say, it is possible for us to organize such programs only if we have extra time. all of us have been same limited period of time, right? Within the available time, we have to engage classes, practicals, correction work, everything. So many times it is not possible for us to organize such extracurricular activities for you students. But whenever time permits, they did. In future also, based on students' interest only, we can organize such programs, expecting your wholehearted cooperation in future. If you are ready to involve in such programs, we will conduct many more such programs. I assure you that we, the faculty members, are ready to serve for you. We care about you students. We want you to succeed in your future and achieve your goals. With these few words, I want to thank all the students for your active involvement, and I also extend my sincere thanks to my colleagues for their presence in this series of programs. And before concluding my talk, I call upon you students to participate in the coming national level webinar that is scheduled for Monday 3 to 4:30. It is for the first time we are organizing a national level seminar. Of course, it is not seminar, it is the online seminar. We call it as webinar. Once few years back, we organized one state level seminar on mushrooms. Later on, it was not possible for us. You may not know, for organizing such programs, we need fund. Many times, due to lack of funds or due to lack of time, it is not possible for us to organize such programs. But in online series, it is a bit easy for us to organize it. Just we have to arrange the resource person, and we should have some technical skill, that is all. We have our colleagues to help in that direction. So it is easy for us to organize for such programs. With your cooperation, we can organize many more such programs. But what is more important is your support in terms of your presence during the program. That is all. Okay. Thank you for the opportunity.
Thank you, sir, for your kind words of feedback. Now, I request Divya Madam to say her opinion.
Just 1 minute. Just 1 minute. Just I want to add
Okay, sir.
just one or two sentences. Some of you might have noticed some harshness. Actually, it is not harsh words. Due to any reason, if I have used any harsh words, don't think it as harsh. It is mainly based on concern, due to concern towards students. I might have used such words, or the tone might have changed. Nothing else. I always like my students. I always try to show concern as well as care towards my students. It is you, up to you to understand it and to harvest it. That is all. Over to you, Divya.
Good evening, all. So, I'm just in a poor network. I don't know how much time I can talk, but actually, I want to share my experience regarding to the apiculture. So, before that, coming to the nature club activities. So, this semester there was lot of webinars which was conducted by this Nature Club Association mainly. But due to my time shortage, I couldn't attend some of them. But this time, even this time also, I was in not in home or college. I was in outside. But at the end, I wanted to attend it, and now I'm in the home. About the apiculture, as the SCB sir told, involve me and I'll understand. Actually, I wanted to follow that. Uh, by knowing some of the people who was doing this apiculture, especially the one person, Manmohan, you may heard about it. He was started this apiculture from after his, I think, 8th standard. 8th standard he has started. Now he's extracting honey for about 150 tons per year. 150 ton honey is to extract ಮಾಡ್ತಾರೆ, and you know the side products of apiculture also. So it is having the uh, families, the bee families they will sell. So for one family they will sell about 1000 rupees, and box also they'll sell about the bee wax, so which is also having very costly about the bee wax. So by looking at all, I wanted to culture the honeybees. So when there, I also went for the training of one day. Again, it is by the Sir Suhas Krishna who gave me the information about the training. I went there and take the training of one day, and I think I thought at that time, okay, I can do this. But after, actually, I took it as November 2020, and I started it. I've taken 2 boxes. After that, I have observed it for 1 month. No changes, 2-3 bites, no problem. After that, from January, I observed about increasing the number. Number also increasing, and they're becoming more and more arrogant. I don't know what to do. But later on, I experienced about their biting. Okay, after taking so many bites, I took the precautions. I purchased some more equipments for the apiculture. After February, I observed that there is a collection of honey. I was so happy that I can culture the honeybees. So after 1 month, I have extracted about only 2 liters, but that 2 liters are much more for me. So after that, I don't know what happens. There was rain of 1 week. After that, it is completely vanishing. The number of bees are coming down, and the eggs are also not there. Again, I did something. I brought the eggs from other hive, but it didn't survive. So at the end, I end up with the two families of honeybees. But I am interested till now to continue in the next season. That's from the November. I didn't lose my hope, so which is very important. So all these topics that you are studying under this extracurricular activities, it will be very important for you. ಈಗ ಎಲ್ಲರೂ ಸ್ಟಡಿ ಮಾಡ್ತಾರೆ, ಎಲ್ಲರೂ ಡಿಗ್ರಿ ಸಿಗ್ತದೆ, ಬಟ್ ವಿ ಆರ್ ನಾಟ್ ಶ್ಯೂರ್ ದಟ್ ಆಲ್ ಆಫ್ ಯು ಆರ್ ಗೆಟಿಂಗ್ ದ ಜಾಬ್. But these are the field where you can get the experience. ನಿಮಗೆ the experience ಸಿಕ್ಕಿದಾಗೆ you can earn from it. So you can become ನಿಮ್ಮ ಬಿಸ್ನೆಸ್ ಅನ್ನ ನೀವು ಎಸ್ಟಾಬ್ಲಿಷ್ ಮಾಡ್ಬೋದು ಇಫ್ ಯು ಆರ್ ಇಂಟ್ರೆಸ್ಟೆಡ್ ಓನ್ಲಿ. ಒಂದೆರಡು ಬೈಟ್ ಸಿಕ್ಕಿದ ಕೂಡಲೇ if you are losing your hope means it is gone. So that much I want to tell about the apiculture, and again I'll obey whatever Ganraj Bhatt says. ಸೊ ನಾವು ಯಾರೊಟ್ಟಿಗೆ ತುಂಬಾ ಪ್ರೀತಿ ಮಾಡ್ತೇವೆ ಅವರ ಮೇಲೆ ಜಾಸ್ತಿ ಕೋಪ ಬರ್ತದೆ. ಸೊ ಅವರೊಂದು ಚಿಕ್ಕ ಪುಟ್ಟ ಮಿಸ್ಟೇಕ್ ಮಾಡಿದ್ರು ಕೂಡ ವಿ ಟಾಲರೇಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಆಗುದಿಲ್ಲ. ಸೊ ದಟ್ ಈಸ್ ವೈ ವೆನ್ ಅವರ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಡನ್ ಸ್ಮಾಲ್ ಮಿಸ್ಟೇಕ್ಸ್, ವಿಲ್ ಕಾಲ್ ದೆಮ್, ವಿಲ್ ಆಸ್ಕ್ ದೆಮ್, ಸ್ವಲ್ಪ ಜೋರ್ ಮಾಡ್ತೇವೆ. ದಟ್ ಈಸ್ ಆಲ್ ದೇರ್. ಸೊ ಥಿಂಕ್ ಇಟ್ ಪಾಸಿಟಿವ್ ಆಗಿ ಅದನ್ನು ತಕೊಂಡ್ರೆ ಯು ವಿಲ್ ಬಿ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಇಟ್. ಸೊ about seminar. It was very nice, very informative. So I think you'll get at least 1 or 2% of knowledge by it. So thank you for the opportunity and all the best for all the for coming the exams. Thank you.
Thank you, ma'am. Now, gratitude unlocks the fullness of life. It turns what we have into enough and more. It makes sense of our past, brings peace for today, and creates a vision for tomorrow. Now, I would like to call upon Ms. Smita AP from 2nd BZC to express our gratitude.
Students, let me interrupt in the middle just before the thank you note. Please, all of you switch on your videos once the thank you note is over. This is for a group photo. This is for the records. Thank you, students. Smita, you can go ahead.
A lovely and a pleasant good evening to all, to our most valued and worthy guests, lecturers, and beloved friends. It's a matter of pride for me that I am Smita AP from 2nd BZC proposing a vote of thanks for this event. I, on behalf of my lecturers and the entire community, first of all, spread my most sincere thanks to the today's resource person, Sri Suhas Krishna Sir, Assistant Professor of Zoology Department, who gave his vital time for enlightening us about hard worker bees from his busy schedule to grace this event. Thank you, sir.
Thank you.
Further, I would like to place my hearty thank to IQAC coordinator Professor Shiva Prasad sir for being in this program for a short while. Thank you, sir. I mention my sincere sense of gratitude to convener of our nature club, Dr. Smita PG. Thank you, ma'am.
Thank you, Smita.
I am very much fortunate to thank our well-wisher, Professor Sri Krishna Ganraj Bhatt, HOD of Botany Department, and Professor Ishwar Prasad Sir, HOD of Zoology Department, for their encouragement in all our efforts. Thank you, sir.
Welcome.
I also owe special gratitude towards Divya ma'am, who will be always supporting us. Thank you, ma'am.
Thank you.
Now I thank our Secretary and Joint Secretary of Nature Club for planning this event. Thank you, guys.
Thank you.
Last but not the least, I thank my friends for your cooperation on this rainy day in making this event a memorable success. Once again, I thank you all for your attention. Thank you.
Thank you, Smita, for your words. The colors of nature is so diverse that it feels great to experience the beauty, and the treasures hidden in the heaven is so rich, precisely in order that the human mind shall never be lacking in fresh nourishment. Thank you one and all.